Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
Okay, today I'm going to model mitosis, and we're going to do it with a simple organism. In this organism, it's going to have, we'll make it 4 chromosomes, and I'll add those other 2 in just a second. And so our diploid cell is going to have a diploid number of 4. What does diploid mean, haploid mean? Well, in this case, diploid means that you have a chromosome from your father that you get from your father and a chromosome that you get from your mother. Now, we would call this chromosome 1, and that's because it's the longest one. If we had 2 other chromosomes, this would be chromosome 1, and this would be chromosome 2. Now in us, we have 22 what are called autosomes, and then we have sex chromosomes. But the way chromosomes are named are just their number. 1 is the longest, 2 is the next longest, 3 is the next longest. So a few other things you should know about a chromosome. This would be the centromere, the center where they're connected, and then each of these beads represents a gene. Now here we just have a few genes, but in a human cell, we're going to have thousands of genes on each of the chromosomes. And so this one right here from dad and this one from mom are what are called homologous chromosomes. And what that means is that they have the same length, but they don't necessarily do the same thing. What does that mean? Chromosome 1 from dad and 1 from mom are going to be exactly the same length. Centromere is located in the same thing. And the genes will be in the same spot. So for example, if this is the gene for blue eyes, this would be where the other gene for blue eyes is. And so you may be asking yourself, well, how are they different then if everything seems to be the same? Well, this right here could be a recessive gene for blue eyes, and this could be a dominant gene. It doesn't give you blue eyes. And so those alleles are going to be found on either side. Or this right here could be the gene for hitchhiker's thumb, makes your thumb bend backwards like this. And over here, so hitchhiker's thumb here, this could be one for a straight thumb, so you don't have a hitchhiker's thumb. What's our goal in mitosis? The goal in mitosis is to make an exact copy of the nucleus. And so this circle right here, bounded by this, is going to be the nuclei, and these are going to be the chromosomes inside it. Now, what does a cell do before it divides? A cell will get bigger, and then it will copy its DNA. So let me model how that works. When it copies the DNA, this chromosome will have an exact copy of it made. This occurs during the S phase. This chromosome will have an exact copy of itself. This one will have an exact copy of itself, and then this one will make an exact copy of itself. And so if this is a gene here, there's an exact copy on this side. In other words, this side and this side are exactly the same. In fact, those are called sister chromatids at this point. And so this would be at the end of the S phase. What happens after the S phase? It goes into another growth phase called G2 phase. So G2 phase, all of that is part of interphase. Now would it look like this during interphase? You wouldn't see the chromosomes during interphase. All of that DNA would be loose within the cell doing its job, the job that it normally does. And so you're really not going to see chromosomes look like this until we get to prophase. Okay, what happens during prophase? All that DNA is going to coalesce and we'll actually be able to see it. Okay, what happens next? So that would be prophase. The next thing is going to be metaphase. What happens in metaphase is that these little spindle fibers will attach to these and they will line up and meet in the middle. And so they're going to line up like this. This chromosome will line up like that, and this one will line up like that, and this one will line up like that. And so the reason they line up like that is that the spindle fiber is attached to them. And they're going to go from here to here and here to here. And then we're going to have attachments over on the other side as well. Now these spindles are going to go outside of the nuclei, which I would draw, but I can't quite fit that on my screen right here. And so there's going to be a tug on either side by the spindles, and that lines these all up in this perfect, what is called the metaphase plate that goes right down the middle. So this would be metaphase. What happens to next? Next is going to be anaphase. And so in anaphase what happens is these are going to be pulled to the side as those spindles shorten. They'll be pulled like this. And these will be pulled. This all happens at the same time. And these will be pulled like this as well. So they're going to move to the sides. And then the spindle fibers are going to start to disappear. And then we're going to have 2 brand new nuclei. So this would be one nuclei, and this would be another nuclei. Now if you look at the chromosomes in each of these nuclei, we've got one like that, we've got one like that, we've got one like that, and we've got one like that. They're exactly the same. In other words, we have 2 brand new nuclei, and each of them have the same duplicate DNA in each one. And that's the goal of mitosis. The goal of mitosis is to make 2 exact copies of the cell. Okay, so I'm going to take a minute and clean this up for just a second, and then I'm going to show you how that differs from meiosis. So in mitosis, we're making two nuclei, and those nuclei are going to be exactly the same. But now let me show you what we do in meiosis. In meiosis, our goal is not to make duplicate cells, but to actually make different cells. We make genetically different cells. And so how does it start? Well, it starts the same way. The cell's going to make a copy of itself. And so it's going to go through a growth phase, so the cell will get larger. It's then going to duplicate its DNA, so it's going to duplicate its DNA like that, and like that, and like that, and like that. So this is going to be at the end of interphase. So the cell has grown, it's copied its DNA, and then it started to grow again. But now we're going to have something different happen. And so in meiosis, we have two divisions. And so during the first division, what will happen is the homologs will come together. And this forms something called a tetrad. And so we'll have this homolog together and this homolog together. In other words, the chromosome that you get from your dad and the chromosome that you get from your mom will actually come together, and they'll wrap around each other like this. They line up along the middle, so this would be in metaphase 1. And they're going to, this actually happens a little before this during prophase, but what will happen is that they're going to wrap around each other so closely that portions of one will switch with portions of another. And portions of this one will switch with portions of another one. And so during prophase 1, they're going to switch bits of them. Or this one right here, it might pop off here, this little gene, and it might switch with another gene over here. So it'll switch like that. And what that gives us is variability. And so this would be during prophase 1, we cross over, and so we switch some of the chromosomes. Another important thing happens at the next step, which is metaphase 1. This might line up like this, but it also might line up like this. And this might line up like this, but it also might line up like this. And this is called independent assortment, as I start to lose one of my chromosomes here. In other words, depending on which side they line up on, they're going to be pulled in a different direction. Now, why is this important? Well, let's say this right here is the gene for Huntington's disease. Huntington's disease means you're going to die when you get to be 40 years old. And if it lines up like this and goes to a sperm or an egg over here, then you're going to get Huntington's. But if it goes over here, you're not. And so this, when you draw a Punnett square, is where that actual genetic 50-50 split takes place. That's called independent assortment. Okay, so now what happens next? We're going to attach a spindle on each of these. So there's going to be a spindle here, and there's going to be a spindle here. And that spindle is going to, during anaphase 1, it's going to pull them apart. So these are going to pull apart, and these are going to pull apart to the side. Now what happens next is we form two new nuclei. So I'll do that like this. So we form two new nuclei. And now the next thing we go through is we don't go through another interphase. And so there's no interphase, but what happens next is it will actually line up again. So it'll meet in the middle, and these will meet in the middle like this. And then it'll simply split them in half. So this one will go this way, this one will go this way, this one will go this way, this one will go this way. The same thing will happen here. These will get split to the side, and these will get split to the side. So what is that? Well, we now create 4 cells. So here's one cell, here's another cell, here's another cell, and here's another cell. So in meiosis, what we create are 4 nuclei. And how is this different from mitosis? Well, we've reduced the numbers of chromosomes. There's 2 in each one. And also we've made them all different. And so if you look at the color combinations in each of these nuclei, it's totally different. And so what do these become?
Adalah pemeriksaan